Talking to Roger, right?
Yes.
The outline that you're going to see on Angel is that outline right there. And they're going to, and the blanks, as I teach, you get to fill in the blanks. My test comes from my outlines. So there's no secret there. It comes from the outline. Now, I may tell you sent me a class in passing that you might see on a test also. But 90% of my test is right here in front of you. So that's 8 right there, if you master it. Okay, that's on Angel. Now it says lesson 13 because I forgot to renumber it, so that should, I will change it to lesson 1 for this new class. But I'm going to change it now. Say no. And then, this is the lesson, this is my test right there, but this is the lesson, and I'm going to change that to a 1. And even all these are going to be changed to 1s also. And this PowerPoint is on Angel 2. So what you want to do over the weekend is fill that sucker in. So when you come to class next time, like well, tomorrow, I guess. Tomorrow, right? Not the weekend. I'm still messed up in my schedule.
Tomorrow's a lab.
You'll have a chance to catch up. Now, how many folks right now don't have this stuff? So I got to go slow for you. Okay.
Sorry.
No problem, no problem. Do you have an outline even?
No, I didn't print it out today.
Fill the write the blanks down. Just copy the blanks and then you'll. Okay. Glycolysis, respiration, fermentation. Let me explain before I even start what we're talking about. Every living thing does this. Everything does glycolysis. Some things do this and some things do that. So after you've done glycolysis, if you have oxygen, then you do this. If you don't have oxygen, you do that. You know when yeast make alcohol like beer and whatever? They do glycolysis, but there's no oxygen, so then they do alcohol fermentation next. But the alcohol is their waste. That's their crap that we want. Now, if you have oxygen, fermentation will never occur. You ever opened the bread up, had a sweet smell coming out of it, and you go, oh, this don't smell like bread, it's a sweet smell. That's fermentation. But when you seal that bag up, what you done is an oxygen supply. You've removed it. And the yeast inside there, they use yeast to make that bread rise, right? Well, that same yeast then start to do the fermentation. And anything wrong with eating that kind of bread?
The sweet bread or the alcohol smell bread?
There's not wrong. A lot of folks they won't do it. There's nothing wrong with it. But anyway, we're going to talk about this one because this one can go this way or that way. I'm going to do this one first. And then do some of this. And when I get done, we'll discuss. Now, fermentation, this happens in your cells too. But you don't make alcohol. You do something else in there. Ever hear of a second wind? And you run and get a second wind? It's involved with a second wind that you get enough oxygen to finally get rid of this lactic acid, and maybe you can run like the wind then. All right, so let's go ahead and talk about it. All right, just copy the red things down, and I guess you too, Roger. When you get the thing off Angel, you can fill them in. I'm going to try to copy the whole slide; it's too big. But that's the number 1, and just say energy, sun, photosynthetic. And I wouldn't worry about the fact that it's 9 lines long. It's not that long on your outline. I'll talk again when I can. I don't want to detract from your process.
I mean, I can just fill them in at home because I got the PowerPoint anyway, so if you just want to go at your own pace.
Well, now, you do know, right, that life on the earth depends on the sun because plants make the food that we all use, right? When you eat a cow, he ate the plant, didn't he? So you get it that way. if without plants, life will cease to exist on this earth. Okay? It says energy, you know, how does a plant actually trap that energy from the sun in that pea you eat, or the apple you consume, or the lettuce you eat? How does it do it? Well, it's very simple. It's called photosynthesis. And what happens, the sun shines on the plant, and the sun's energy is used to take chlorophyll, which you saw last time, excite it. It gives off electrons, and then those electrons are then used to make energy. And then that energy is then used to take the carbon dioxide the plant took in, right? And the water he took in, and make those come together to make more plant products. The black-eyed pea you consume, that's how it's made. The flower that blooms, that's how it's made. The root that cracks your driveway, come into the surface to find water. That's how, and they're strong, as you well know. They'll lift the house foundation and crack your house. That's how strong a root is. But down here, here's the problem. I cannot use that food I've eaten if I don't have some way to trap the electrons I'm going to get from it. It's like working in a cookie company and they're getting by you and you can't catch them fast enough. They're wasted, right? Well, with glycolysis going on, do you know what's our main fuel? What do we want most? What kind of sugar do we need? It's the G word. Glucose. Glucose is what we get from the black-eyed pea, the sweet milk we drink, the bread we consume. It just comes from everywhere. And that's our fuel. Even if you take in a lipid, a fat, you can change that to glucose. But the problem is, it's hard to change. So what happens, if you're eating carbohydrates and fats, and a lot of fats, if the carbs are enough to maintain your life, then you're going to save the fat for a rainy day. On your hips, on your thighs, and it never rains. And you keep eating. So what do? You get fatter and fatter. At what point how come a person can't be 2,000 pounds? At what point does it stop? When they die. When you can't no longer sustain your girth, and you die. And you know overeating is a killer. The problem is it's just so darn fun. All right, so it all boils down that the plant takes the sun's energy and traps it, and those products it makes. Okay, we need that sun's energy to make my hair grow, to make my muscles work, to make my heart beat. And the question is, how? I can't. That sun don't bother me. It doesn't make vitamin for my skin. But I got to have a way to get the sun, so I eat the plant. Or I eat the cow who ate the plant. And then I get in me those products the plant made. The question then is, how do I get that sun's energy back out of that stuff so I can keep living? Well, that's what glycolysis is all about. We actually are able to take the glucose, split it in half. And when you split it like a firecracker going off, out comes the energy that I can use to make me live. But when you stop eating, of course, you lose weight. But you start consuming your own body parts. And then next thing you know, you die from some disease. But anyway, that's the point of the whole slide tells you this. It all starts with the sun. And the sun's energy must be trapped. Animals cannot trap it. Only plants can. They're called producers because they produce the food. Now, if you're a vegetarian and you never eat meat, is there a problem? No. If you're carnivore and eat tons of steak, is there a problem? No. Either you eat the plant and get the energy directly, or you eat the cow. But what's the better diet, vegetarian or carnivorous? Because the carnivorous diet, you're taking in fat too, right? Those saturated fats.
Lord, your sister.
like lard and the Crisco. The ones you pour, like the oils, those unsaturated, those are good for you. But you know, the old white lard, you drop it into your fried skillet and you melt it and fry that chicken, tastes so good. The oil ain't good for you, but the chicken tastes real good, and that's why a lot of folks don't like fried chicken. Personally, I like fried chicken. All right. Now, we have got to have some way to give off electrons or protons. And I didn't say protons because actually it's hydrogen. Because hydrogen is one proton. If you have one proton, that is hydrogen. So I use this symbol, actually I did, everyone does. Electrons and protons have to be donated because when you give those off, that's how you're going to get the energy back. But the problem is, I got to have donors. Well, you know that these pigments in the plant, when the sun shines on those pigments, they throw off electrons. They donate electrons. They give off the hydrogen ions, the protons. Glucose can also be a donor. You do that yourself. When you break it down, out comes the electrons. Now there's some things that live in sulfur that would kill you. For them, they use sulfur as their donor. Their metabolism is different. I mean, question, why is it that a deer can eat that mushroom that would kill you if you ate it? Why is that? Or think about things a buzzard eats. If you eat those things, you die. Why does a buzzard not die?
Because the system is
It's the
different.
metabolism. You got it right. The system, the metabolism uses the product. If your metabolism, okay, it is your metabolism that determines what's good for you, what's bad for you. And their metabolism can utilize it. Yours cannot. What won't kill them will kill you. It's all metabolism. You taking this sulfur and kill you because that's not your metabolic way. Ammonia. You smell ammonia? There's some things that live off ammonia. You ever open a diaper pail and smoke that come out of there? How strong the bacteria, and they love that stuff. You think ammonia would kill bacteria? Some bacteria thrive in, I thrive in oxygen. They die in oxygen. There are some bacteria that cannot live if you give them oxygen. They're called obligate anaerobes. An anaerobic thing means don't give that sucker oxygen, you're going to kill them. And if you now, some anaerobes can take or leave it. Some anaerobes say, well, I don't care if I have oxygen or not, it won't hurt me. So if you get my fine, I can use it. Don't fine, I can use it. But there's some organisms that are obligate anaerobes. That means you can't give those suckers oxygen or you will kill them. And if you've ever had the old cloth diapers, when we first had our first child, we were trying to save money, we used cloth diapers. Good God, I never tried that again. Our second boy, we bought Pampers. And I don't know if it's cheaper or not, but I know that that little white diaper pail in there, after a few days, and my job was to get the commode and kind of roughly clean them, chuck them in there, and then go wash them. God, that was terrible. Hydrogen gas will provide electrons, and some things can live in here. But here's the trick. Whatever it is has got to give up electrons, because those electrons are used to get energy that you're going to use to build an ATP. You must have a supply of energy so you can build ATP. Electrons flowing. If I had, you know, little fans you blow and they spin, kids pulling bicycles. Okay, if that thing, say that's a turbine, a wind turbine. If it spins, can it make electricity? How about if it doesn't spin? Well, if my wind is the electrons, and I can spin this machinery, it can make ATP. And as long as I got electrons, I can get them from here, that's the wind, or from here, or from here. And the kind of metabolism you have, depending on where you get that wind from, if you can spin that little turbine, you can make the energy. Some folks use oxygen to spin it. Some use glucose to spin it. If you spin it, you're good to go. My problem is though, you know, if I try to live off ammonia, I'm going to die. My metabolism can't use ammonia. But we call these things acceptors. Now, let me back up. Not acceptors. When I give off an electron, something's got to capture it to use it in the next step. It'll get away. It'll get away. And the things that grab it are called acceptors. So let me start again now. Let's look at chlorophyll. Sun shines on the chlorophyll. Chlorophyll throws off electrons. Something's got to grab them, or they'll get away. So the chlorophyll is your donor. But something's got to say, and grab them, and then use them in the next process. The grabber that grabs them is the acceptor. You have a donor that gives off the electrons and protons, and you got an acceptor that says, no, you ain't going nowhere. I can use you next. I'll use you later on. Otherwise, it's gone. Your heat. You're warm right now because you flat cannot capture all the energy you give off. Energy is heat, right? You ever have a light bulb burn out and you grab it too soon and you burn your fingers? Now, those CFL bulbs, they can be hot. You can touch them. But incandescent bulb, the old-fashioned kind, if you're reading, been over 2 hours, it burns out. Oh, grab that sucker right now. Put it like a kitchen mitt on. But a CFL light can burn for hours, and you can touch it. It's warm, but it won't burn your fingers like that incandescent will. And the thing right here, all the heat being given off, something must capture it. If you stand by fire, and like most problems in a campfire, you only warm half your body at a time. You know why, don't you? I got to spin around, get my back. I got to spin away because the fire don't get around. It radiates. Well, how about the heat going by you? Who gets it? Nobody does. So in this case, as you stand there, you're the acceptor of that heat, but light's getting away. Now, if I have a friend, he's getting accepted, and the more acceptors you have, the more you can capture, and the more efficient you're going to be. Now, my next slide is about the acceptors. There's two very common acceptors in all living things. Now, one's called NAD, and actually it's nicotinamide adenine dinucleotide. That's why I say NAD. But it's a good name for it. Di means what number? 2. Okay. Do you recall adenine from our discussion on DNA?
Yes.
Adenine always hooks up with thymine. That's the same adenine. Mm-hmm. That's the same adenine and ADP. And same ATP has 3 phosphates. ADP has 2, and AMP has 1 phosphate. He's an acceptor. Now, if you look carefully, you know, I got two nucleotides. That's from this word here. Adenine is a base. They're bases, okay? Now, one of the nucleotides is this thing, which is actually niacin. Vitamin. That's why vitamins are good for you. You need vitamins, right? That's the reason. One of your acceptors going to help you make energy contains vitamin B3. You want that in your diet because you need to build the NAD without vitamin. You won't build it without niacin. Now, when it's not carrying it, it's called this. When it's carrying hydrogen, it's called that. NAD is prepared to carry. This sulfur is loaded. And there's what you carry. That H that was given off when the sun hit it, that's how you grab it. It'll get away and be gone and you can't use it. So this is a carrier. Now there's another carrier called FAD. Flavin comes from B2. Adenine, that's the same step you have right there. It's the same base. dinucleotide, there's not really two nucleotides, it's only one, but the problem is we found this one first. And so we had NAD, we just called this one FAD. Even though D is really wrong because it's not really a dinucleotide. It's a misnomer and it's stuck. And we've never changed it. We just tell people it's not a nucleotide, it's not di. It's just that if we had found this one first, we probably would have called it something else. But we found this one first. It's called, he was NAD, an acceptor. Let's call him FAD. Then we realized that that D is misleading because really and truly it's not. Now, when you're carrying, he can carry two. He can carry two H's. He can just carry one. So who's your better toter? NAD or FAD?
NAD.
Huh?
What they call it?
FAD.
Yeah, he carries more. Yeah, if I can get one, if I can get two for the price of one, wouldn't you do it? But this is the more common. You see more of this stuff because it's limited. You don't see much of that because it carries so much what it does. If I had 100 of these, I expect to have maybe 50 of those. You know why I said that? Couldn't 50 FADs carry 100 hydrogens?
Mm-hmm.
No, they're about the same. That's why I expect to see more NADs than the FADs. And you're going to see more about this later on. But I want to tell you, you've got to give off the electrons and the protons. And you've got to have a way to capture them to use them later on. And your two biggest capturers, NAD and FAD. You're going to see them in design that detail later on. Now, the next part of PowerPoint is just going to talk about one thing, glycolysis. And every living thing, I don't care what your metabolism is, you do glycolysis because it doesn't use oxygen. Okay, so let's talk about it. Pretty simple. You do the energy stay alive, right? Okay. The first step of getting the energy is the thing called glycolysis. Now look down here. Glycolysis, this process splits glucose. See the GLYCO? That's the Latin derivative for glucose. Lysis, the word lysis means to split. It means to split. So if you translate glycolysis, it really says splitting glucose. You just cut it in half. Now, if glucose itself is made of 6 carbons, and you cut it in half, you got 2 brand new compounds, right? How many carbons each? 3. There's no oxygen used in this process. It's enzymes that do the splitting, and ATP. Now, so this doesn't matter. It can be there, it cannot be there because it doesn't use it. So oxygen is not a factor in glycolysis because it's not used anyway. It only happens in the cytoplasm, not the mitochondria. Remember we talked last semester about the mitochondria giving you energy? It does. But glycolysis does too. But that's not in the mitochondria. That's in the cytoplasm of the cell. And like I tell you, glucose is 6 carbons big. And if I split it, I get 2, 3 carbon. Now, I call it pyruvate. And some folks call it pyruvic acid. Same thing. So 2 pyruvic acids come from splitting of 1 glucose. So when you're done with glycolysis, what do you have? 2 pyruvates. Well, you had 1 6 carboner, then you got 2 3 carboners. Now, think about it. I've got to use 2 ATPs to make that glucose split. Follow me so far? So, if I want to hire you to do this, I'm going to give you $2, and you're going to split the glucose in half. Now, when that's done, you can then give me back $4 because those 2 pyruvates can make me $4. But how much am I up? How much am I up by? What's my gain? I gave her $2. Give me back $4. How much have I gained? Just $2. Now, did I gain them?
Yeah.
It took 2 to make it happen, and she gave me back 4. So 2. Now I got nothing for. I gained 2 ATPs. That's what it's for. It's just a little gain, and when you're running, you go fast, and you can't get enough oxygen, and your side starts to hurt. It's because you are doing glucose, and you're making all these acids that you can't get rid of. And you got it's called oxygen debt. When you get done running, you're going, you're breathing, because you got to put back in the oxygen to get rid of all the pyruvate that you produced while you ran. You will slowly recover. And I've always had high school coaches, when you run, walk it off. Don't lay down. You heard that too? Why do you not just crash and lay down? Some folks do. If you walk it off, what's that blood doing? Circulating, carrying oxygen. If you lie down, your heart's going to slow down. That's going to slow down the recovery process, and you're going to be taking a long time to recover. If you walk it off, that's good exercise. It didn't say run it off, walk it off. It's always good. So the thing about it is now, let's say I have 4 ATPs to spin. I give her 2 of them to make the glucose splits. Right? She gives me back how many? 4 ATPs. Now, I gave her 2. I had 2. Gave me back. Now I have 6. Have I gained? Yeah. If I have $4 in my hand, and she can do something, but she'll do it with $2, so I'll give her $2. She does a thing, and I then give me back $4. The 2 I had, plus the 4 she gives me, I have $6 now. You follow me? I had 4, right? Now I got 6. I'm up by 2 again. It's always up by 2. Now, if I gave her 4, she gave me back 2, how good a deal is that?
You lost 2.
I'll go out of business. I will die. I got to give her less than she returns to me. And glycolysis does that. This is good in a pinch. If you want a lot of energy, you supply oxygen. You'll get tons of money back. And that way, when I give you $2, you give me back $36. If I use oxygen, is that a good deal?
Mm-hmm.
Yeah, because if I use oxygen, for my $2 I gave you, I got 2 back from you. I got 36. I'll have $38 in my hand. Oxygen is always the best way to go. But if you're running and sprinting and you just can't provide enough oxygen, you do glucose, you do glycolysis. And because you can't keep living that way, you stop, you go, my sides hurt and everything, you know how it feels. All right. Now, here's what happened. Did you know from last quarter I told you that all these things are split by adding water? You remember that? How you add water and make things come apart? And how do you make them come together? Pull out the water, remember? If I want to take that glucose and make it go apart, I got to provide water so it can separate. That's why you drink tea while you eat. If you're not, you're going to get real thirsty. Because you're going to use that water from your blood. Until your mouth will dry. That in turn gives you a sense of thirst. Now, like I told Hillary, when that water splits it, she's going to give me 4 ATPs. I didn't use 2 to make it though. So, like I said, I'm going to use 2 to make it happen. So now I'm broke. But when it happens, she gives me back 4. Now I ain't up $4, am I? I'm up only $2. If I keep doing that, I'll keep slowly building my bank account. But you give me oxygen, I can make a lot more at one lick than just two at a time. And by the way, glycolysis, this glycose was what we used to call glucose. That is the old school name for glucose. This is how we got that word. And lysis, as you know, means to split. Hydrolysis, what's hydro mean? Water. So you split by adding water. So that's the term glycolysis. It just means split glucose. And in doing that, I'm going to gain some ATP. Not a lot, but enough to keep me living for a little while. Now, if you're a one-cell animal, that's all you need. Now, if you're a little yeast, two, man, you're high on the hog. Big as we are, though, we need more than two. But a little yeast can do that because a yeast doesn't use oxygen. It gives off carbon dioxide, and that forms the bubbles that make your cake rise, that make the bread rise. Why does the bread collapse when you open the door too fast on the oven? What makes it collapse? Ever heard about one collapsing? What makes it do it? The air pressure change. When that cake is baking, it's moist, right? There's not much holding that thing up. You try to put icing on and tear your cake up, try and do it. And the cake is so fresh, and the icing just pulls apart. Okay, wait a little while. My father used to work at Colonial Baking Company. And I'd go in there, those floors are so slick. From the flour on the floor, I just slide across there. Boy, don't do that. I just slide back and get spanked. But anyway, that's the whole thing about glycolysis. It's good for a pinch. Every single thing does glycolysis. Now, whether you have oxygen depends on what you do next. Okay. Now, this is the formula. There are several of them. This should be one long line. This is on one side of the line, so if you can, make it one long line. Because right here is one long line. I made it big for you. But there's your glucose. There's your adding the energy. It's going to split to 3 carbon pyruvates. Give off some water, that's what makes your bread moist. And enough energy to make 4 ATPs. Well, I used 2. I made 4, so I only gained 2, really and truly. All right. And this is a little bigger so you can read it. And now, see that C6H12O6? Are there more things than glucose for that formula? Fructose is. I must write the word glucose under it because it would be confusing because that formula applies to fructose and galactose and glucose. All 3 of those have the same formula. That's why your body can take fructose and change it to glucose and then use it. Because it's the same formula. It's just they're added or rearranged differently. So that's why I always write under here. And there's more things than pyruvate that can be C3. The 2 means I got 2 of the molecules. There'd be a 1 right here. So one of these, if I cut it in half, there's your 3 twice, right? I got 2 pyruvates. Now, water's going to come off in the process. But this is what I want. I want the energy. I want the energy. Because now I can make myself 4 ATPs, and it costs me 2. So I'm $2 ahead. If I keep doing that, I'll be another $2 ahead. And I was thinking about it, I need the energy. And I can't even capture that without ATP. ATP catches my energy for me. That's my storage facility. You store everything you have. This is a diagram I found that shows, it's pretty good. I went looking for a good diagram that would explain what's happening, and I found 2 of them. that were different, but they were trying to show the same thing, and I liked it. On this one, there's your 6-carbon glucose. There's your ATP coming in. ATP coming out. The ATP adds, see that phosphate right there? It's going to be tacked on here. That phosphate makes this thing unstable. If you're unstable, what happens? You break apart.
Okay.
So when they break apart, you have these 2 3-carboners. But these 2 are not those 2. Because I want to take, you see this comes in without the hydrogen and leaves with hydrogen. See it? So this thing has 1 more hydrogen than he does. If he's got 13 hydrogens, how many has he got? 12. They're both 3-carbon compounds, right? That's why I wrote the word, now, PGL and is phospho, I can't say the word, phosphoglyceraldehyde. The only difference in this one and this one is that he has a hydrogen, he throws it off, he is missing it. I need this H later on to make some more ATP. But I got a way to catch it, and there's the NAD. There's my catcher. There's my catcher. Otherwise, this thing be gone and the room would be gone. I got to catch it. So on either process I use, let's look at. I take my glucose, 3 carbons. I input my $2. The phosphorus is tacked onto the molecule, make it unstable. Water is added, it flies apart. Then I have PGAL. This thing is going to throw off a hydrogen. I don't want to get away though. So here comes NAD, a cage to trap the hydrogen. Now it's got hydrogen to use later on. This PGAL then, missing a hydrogen, becomes this thing called pyruvate. The only difference is the number of hydrogen atoms. He's got 1 more than he does. Does that make sense? Now, the question is look here. Here comes 2 ADPs over here in phosphorus. And when this thing is just thrown off, energy is released. And energy is used to take 2 ATPs and make 2 ATPs on both sides. So there's your 2 went in. What's 2 plus 2?
4.
And that's my 4 that come out. I used 2 to make 4. And these 2 diagrams are the best I saw. I looked all over the place where I could do diagrams. When I made them bigger, they didn't get all blurry. But it tells a story. Now, this pyruvic acid, if there's oxygen, something happens to it. If there's no oxygen, something happens to it too, called fermentation. So this is going to be a start of either respiration, or it's going to be a start of fermentation. If you have oxygen, this is going to start respiration. If you don't have it, it's going to start fermentation. That's why when the thing first started, I tried to tell y'all that this starts everything. When you're done, you have pyruvate. Now, if you have oxygen, the pyruvate does this. If you don't have oxygen, it does that. But it all starts here. It all starts there. Now, and it looks like I misspelled something. Oh, that's aerobic. 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 And the word aerobic means what? With oxygen. Anaerobic means without oxygen. Okay. Okay, so we're going to take a look at And I'm going to come back and do a better job. I want to take just a brief look at what happens if you have oxygen. Okay. And then I want to describe the Krebs cycle in more detail later on. I want just to give you right now just a glimpse. This is what you're doing right now, breathing like you're doing. You're doing aerobic respiration. It's the best. It's the cream of the crop. Okay. Now let me back up. Question: in that cell, where did glycolysis happen? In the cytoplasm. Now, if we go any further with this thing, the rest of it's going to happen in the
Mitochondria.
Mitochondria. Because the oxygen goes to the mitochondria. And so does glucose go there, or the pyruvic acids go there? The acids go there. The glucose is gone, right? So the pyruvic acids, which came from the glucose, they go to the mitochondria too. And then they're exposed to oxygen, and they do a number of process steps that give off 36 ATPs when it's done. That's tremendous. And that's why right now you're breathing, easy going. You feel how warm you are now? Because you can't capture all the heat you're giving off. If you capture all, you probably make 100-something ATPs. But it gets away from you. I mean, you're warm because you just can't capture it all. But you capture more than you need. Okay? Now, so glycolysis has happened. What time is it? I'm going to go over time. Okay. We got what, 2:15? Okay. So, in the cytoplasm, glycolysis happened out there. I split the glucose. I gained just 2 ATPs. But now I have 2 pyruvates. Pyruvic acid, right? Now, if I have oxygen available, those pyruvate acids can be used to make me a ton of money if they go to the mitochondria where the factory is. So what happens, I breathe the oxygen in, it goes to the mitochondria. The 2 pyruvates, they go to the mitochondria. And in the mitochondria, there's 2 processes. There's one called Krebs cycle, which is totally different from the ETC. If you take a tennis ball on top of a staircase and shove it, and it goes down the staircase, will it always have the same degree of bounce, or bounce higher and higher and higher?
Higher and higher.
Higher and higher and higher. As you go down, you're going to see it bam, bam, bam. Well, that's how the electron works. As the electrons move down this stairway, the staircase, they give off more energy, and more energy, and more energy, and more energy. And you're making ATP, you make ATP. Krebs cycle is good, but this sucker here is where you make all the energy. He makes, I think he makes 2 ATPs. He makes 34. Because every time the electrons fall down that stair step and they bounce a little higher, they give off more energy. Bam, no more energy. Bam. And that's called the electron transport chain. When you see it, it's like a staircase. And it's chemicals. And as you go from down the staircase, and the more steps you have, the more ATP you can make. And all this happens in the membrane of the mitochondria. You know how the membrane inside the mitochondria, how it goes back and forth? Those membranes inside there is where the ETC happens. But if you go in the juice of the mitochondria, that's where Krebs happens. All right. Now, I'm discussing these 2 mechanisms right here. Both mechanisms make lots of ATP. And all this stuff you know, what's 18 times 2?
36.
Well, this process can produce 18 times more ATP than you did with that measly glycolysis. You get 36 ATPs. Now when you add back the 2 you already got, that's your 38, right? So one glucose molecule has the potential to make a grand total of 38. 2 from glycolysis and 36 from
86.
the Krebs and the electron. Now, but when you do this, you're making trash. If you go, what'd you breathe out? That's one thing you're producing. When you break down the pyruvic acids and you release all this energy, one of the things you're going to make and get rid of is water. You breathe it out. And in cold weather, you see it coming out. That's coming from your lungs, which come from your blood, which come from your cells, which came from the mitochondria. And you see it go out. I mean, I do this. You see the vapor?
Mm-hmm.
That is from respiration. Now, carbon dioxide, when I breathe in the oxygen, okay, and I split the glucose. Now, what's glucose made of? Got some carbon inside it. Got some what else? Hydrogen. And what else? Now, the carbon and the, where's that picture at? I want to find, okay, let's see. It's right here. These two, C6O6 only give you CO, wouldn't it? But what are you breathing in? What are you breathing in? Right now, what are you breathing in? What gas? Oxygen. When you take C and an O and add an O to it, it becomes CO2. And what do you breathe out? Well, the C came from the pyruvate that you broke down. One of the O's came from pyruvate, and the other O came from what you breathed in. Now, the water you produce. Well, you see the H right there? That's that H. It always be gotten rid of. So when I break this sucker down, the C's and O's are going to be joined to oxygen. You're going to breathe out carbon dioxide. But how do you get rid of that H that you had? Well, oxygen you breathe in, it's going to join here too. Every oxygen can hook on to these bad boys, and you get to get rid of the H's that way. So, on the CO2, one of the oxygens you breathe out, what you breathe in, right? And on the water, one of the oxygens you breathe in can turn off 2 H's. When you get done, there's nothing left but these two things. All that's left is water and carbon dioxide. I breathe both of them out. And the pyruvic is totally gone. Because the C's and O's in the pyruvic is in the carbon dioxide. And the H is trapped in the water. Pyruvate is gone, and you have all the energy you have for making it. To me, that's cool. Now, for a long time, I couldn't understand all of it. And right now, for once, I got it. And it makes perfect sense to me. I said, that is so cool. And when I'm telling you this, I'm not memorizing. I am just saying now, here comes, you eat the glucose. It goes into your cytoplasm. You're going to use 2 ATPs to split that sucker in half. That's called glycolysis. And you do that, you're going to make 4 ATPs. Now I'm up by 2. I can do it again. Now, I have oxygen available, so I'm going to use the oxygen to take my pyruvates and just break them suckers all to pieces. So I got to send them where to do that? To the mitochondria. So the pyruvate go to the mitochondria, oxygen go to the mitochondria, and then the Krebs cycle, I'll show you later the Krebs cycle. The Krebs cycle is doing this thing, the electron transport chain doing this thing, just breaking this stuff all to pieces. The carbon, they're dismantled. The carbons are released, the O's are released, the H is released, but you got to get rid of them. And when you get rid of them, is what you breathe in. The oxygen that you breathe in is tied to the C's and O's to make carbon dioxide, and you breathe those are gone now. And some more oxygen you breathe in is tied to those 2 hydrogens you got rid of to breathe those out. So when you get done, everything's gone. But you have all this energy that you have trapped in ATP. That to me is a marvelous thing. And the question, evolution, how did that just evolve into being you reckon? Like somebody planned that. That's all I say about that. The more I learn, the stronger my faith becomes. That's just me though.
There's not many biology teachers that think that way now.
You agree or disagree?
I agree.
I got no problem.
Yeah, most biology teachers, you know, they.
I know.
You know, so.
I know. I mean, I'm fairly intelligent, and I see this stuff, and I go, how in the world can this evolve? Just like somebody designed this and dropped it in place. Now, if I would like to see an environmental biologist explain that to me. See, he can pull BS on some of y'all, but talking to a fellow scientist, that I can say, then explain the glycolysis. How'd that come to be? And a lot of times they'll call your names in. When they can no longer win the argument, what they do, they call your names. That's when you know you won. But you get mad then because you're insulted. As long as he has the argument, he can argue with you. But when you call him on it and he can't, then he calls you a bigot or you're stupid. No, just explain what you mean. Put it in words for me. And if you can't, then I won. But they don't admit that. Now I'm not waiting for it because this is a good punch start.